Okay, so I want to just open that up. Any questions? Right, thank you.
So maybe what I would just ask is for all of you to just, so maybe Corey and Mark just ask you first. This program's totally dependent on having. Sorry, I'll address this to Corey and to Mark. This program's totally dependent on having a user partner. So tell us about your experience in securing a user partner, and just advice to this audience on the way you went. I mean, was it just natural that you went to the BC Pharmacy Association, that you went to different partners? I mean, how did you do this, and what advice would you give to the audience?
I can take a stab at it. It was natural as soon as we both got in the room together. So I've been doing, as I think everyone in this room is familiar with genomic evangelizing, and so I had my elevator pitch and have been giving it to a couple of groups. But as soon as we were in the room with the BC Pharmacy Association, it was obvious that we both had shared interests. And the second part of your question, I know, is the important one. What was that again?
Yeah, it's just the advice you
Advice.
Yeah.
So advice is, for me, is treat this as a long-term relationship from the get-go. There were bumps along the way. There were disagreements where I wasn't sure we were going to get through them. And having come through the other side, as I mentioned in my talk, I've learned an enormous amount about compromise and having the big goal in mind the entire time. And in the whole process, Genome BC was really a coach for us to get to this point. So, yeah.
Mine was slightly different. The user is the patient. So that's the final stage. So I had to sequence down to, that's the wrong word to use in this audience, I had to work out who would be, if you like, the commercial user, or the person who actually puts this into place. So it's not the family physician or the pharmacist who puts this into place. So we backed up a bit, and it's the electronic medical record people. They have to be the ones who are engaged. I worked with one electronic medical record company and they were fine with it, but TELUS is bigger. And I have to say, going to that first meeting with Brendan Byrne, I didn't know how we were going to come out of that. Whether it was going to be, this is rubbish, this is futuristic, this is not relevant. But actually when we did our pitch, and yes, we've done the same, lots of practice, incredibly well received, almost overwhelmingly received, and suddenly we were back at Genome BC saying, oh, we've got one, this is who we're going to work with. So, yep, still, I mean, after that enthusiastic reception, lots of road bumps, and we have to work through that relationship, and it definitely is long term, absolutely.
So I'd actually like Brian to answer that question too, because one of the things I've learned in this process is that selling tree seeds is not quite as straightforward as one would think. So I think this question is actually quite relevant in that sector too.
Okay. So it's with regards to, can you hear me?
Yes.
Okay. About the user. Well, the primary user in our case is the Ministry of Forests and Lands and Natural Resource Operations, because it's our tree breeders that will be directly applying the results of the genomics. To attract private investment in this research is difficult because, as I mentioned in the talk, 95% of the province is publicly owned. And so the forest companies are tenants on the land base, and they have obligations to reforest, but they do so at, they're using ecologically appropriate species and methods and employing professional foresters to manage the forest. But they firmly believe that it is the province that needs to invest in the land base, and their job is to look after it. So their obligations are to reforest, and then only to manage that stand up until it's free to grow, which can take 5 to 15 years. However, the companies that I showed you on the slide there, 2 of them operate almost exclusively on private forest lands, and they have some of the fastest growing sites in British Columbia. We're not as productive as Washington, Oregon, that have a lot of that deep ash and very nutritious for trees. But yes, they're harvesting trees that are 35 to 40 years old now on private lands. And so if there is opportunity, so those are the groups that I think could see potential opportunities for investment in genomics. And I think Weyerhaeuser south of the border is actively pursuing this as well. So that's a little different than some of my colleagues here, or particularly in the agriculture industry, where you can get a turnover of your breeding cycles rotations, whether it's an agriculture crop 1 year or if it's dairy cows in several years. So you can get your return on investment. It's a different situation in forestry here in British Columbia.
Any other questions? Marty?
Hello, this is Marta, and I work with Martin. And I have a question for both Martin and Corey. I wonder if you can tell us what your thoughts are in terms of complementarity of these 2 approaches, of the pharmacy and the clinician. I think most of us have either experienced or have seen someone experience either a drug that just did not work or a drug that brought a kid to the hospital. And at this point, we have no way to easily use the pharmacogenetic information to prevent that. So it seems to me that both approaches are absolutely essentially, urgently needed. Any approach that will bring us the possibility of having pharmacogenetic information to avoid this or increase safety and efficacy of the drugs. So to me, these are complementary, and just we need to try them until something happens. Could you comment on how you see these different approaches complement each other, or even parallel at one point?
So very good question. There are several elephants in the room when it comes to human genomics. One of them is who and where is your data stored. So who stores it and who has access to it. One other, because there are many, one other is when you do the tests, how reliable are they? Are they actually measuring what they say they should be measuring? And I think both of us are dealing with those problems. And there isn't one solution we can just pick out and say, this is what Oregon has done and we can use it, because no one has done it. UK, other countries in Europe, other places in North America, they're talking about biobanks and they're talking about how, but this is about clinical use. This is everyone having genetic material stored and accessible. And I think I don't have a solution where we're going to end up, but I think both our projects, if you said, where is the overlap? I think that is one major part of where we really work closely, and we've been looking at different solutions and teasing out strengths and weaknesses of both approaches.
Yeah, not to be too agreeable, but I definitely agree that this is a big problem, and it's, as I evangelize and try to say, this is the year that we actually roll up our sleeves and start to see what's going to work. There's, we need many shots on goal. And I think one of the most important things is that in a group like this, we all can feel that there is going to be a benefit. But in order to convey that and also demonstrate that, it's equally important to get over the fear. And part of getting over the fear is conveying the degree of risk. And I like to say that there really is no risk. And so once we can convey that, we can dampen down the fear. And the whole idea is that if we're both attacking from two different perspectives, at the end of the day, we'll be able to define what works and what doesn't work, but we will have data to support those conclusions. So I think they're absolutely complementary.
Security on the data is one of the most amazing things you're going to have to deal with, I expect, because you can imagine what corporations want to get a hold of, because if they have a genetic predisposition, say, to breast cancer, they'll screen that, a corporation would screen that person out for fear.
Absolutely, that's the fear issue, but sorry, just real quick, is that do you want that data going to a broker that's going to sell it to a pharmacist, or to a pharmaceutical company, or do you want to own the data in collaboration with your pharmacist?
Oh, I would love to own the data, obviously,
Yeah.
but that doesn't mean that we can look at all the break-ins on Home Depot.
Yep.
That sort of thing. So the security is, that's a huge issue.
It is. It was interesting talking with the insurance company, Standard Life. They actually said that the genetic information is basically not going to change them at all, because they know how many people are dying now. They know the age of death of the people, and they provide for that. So if you've got an increased risk because of a genetic factor, why bother increasing the payment for that individual? They say that's too much work. Creating a new system of payment for each individual based on that's a nightmare for them. It's much easier to say you're 45, here's the standard rate, bang, done. So very interested to get that discussion going with the insurance companies. It's not that necessarily, but I do feel that other people would like to sell to you on the basis of knowing what your risk is. They would plug into that, and that worries me.
I'm from the States, so that whole issue of non-insurance is there. Unfortunately, we live in Canada, and that's not an issue.
The other group that help us is InfoWay. This is a group in Canada who organise informational transmission of data related to health, and we're very much plugged into this group because anything that's going to be taken here in BC needs to work across in Ontario and wherever, so the transport of that information is very important. We do have good organisations, it's just a matter of us plugging into those people and hearing what they say.
Question for Marty. So Marty, one of the things that has really impressed me, I mean, if you look at the dairy cattle industry and you look at the selective breeding of traits and how good you guys are at this, it's staggering. I mean, particularly those of us on the human, medical side of things. And so, but one of the things, thank you, that dawned on me is that, so almost all cattle in this industry are the result of in vitro fertilization. I mean, the
Artificial insemination.
Artificial insemination. Artificial insemination. So you guys have an unbelievable. I mean, so can you just comment on that? I mean, just how that, because it would seem to me that when you test it 4 days and you can predict how well a particular heifer is going to do, you have the ability to select those out quite effectively.
The animal agriculture industry has put a lot of money, especially in the United States, on developing the genomic patterns of dairy cows. And we have lots of dairy cows, but mainly Holstein. Now, if we go into the Jerseys, the reliability is not as good. We go to the other breed called Ayrshire, which is a very rare breed. The reliability is not that much improved. So it's the amount of data that we actually have to start with our foundation. And they've been doing that testing initially with the bulls that used for sires, because if they tested good, they can sell more sperm. But interestingly, when genomics came in, they had a lot of tested bulls out there, and one group that did not adapt initially decided, well, we better test our bulls to see how they matched up to what the progeny test was. And so the genomics didn't match up pretty well with what the progeny test was. The progeny test takes about 10 to 12 years for it to come to fruition. By that time, the bull's dead. Kind of hard to make selection on that. Now with genomics, we can make that selection in 4 or 5 days, and now we've advanced genetic selection to the fact that at 10 months of age, we're taking the next genetic strain from the sire of the sire, the son of the son of the son of a proven sire, all genomically selected. It's a bit it's a whole shift of thought. And the farmers have come alongside of it, and they're all wary of it, and they shake their heads and say, how do I make selections now? And I say, well, I have to be the coach all of a sudden. So right now we're the beneficiary of like 10 to 15 years of hard work and data accumulation. And we're fortunate we have a strong associations in the dairy industry, both in Canada and in the United States, where pedigrees have been kept for years and years and years, and the data for milking has been kept for years and years and years to form the basis of the genomic evaluation. So the SNPs can be evaluated, much the same as what he's trying to do with the trees right now, but he's got 10 million trees to be sampled to get an idea of what the SNPs look like. We already have that in our hands. And just so you know, the turnover time is maybe 2 months from testing to results. We're probably going to get down to about 1 month. So it'll be interesting.
Robert?
I was wondering if the panel could comment on the amount of sequencing you expect to do in these different situations in the next couple of years. Are we talking SNPs or gene panels or really big gene panels or exomes or whole genomes? And how do you feel about collecting data now that's more than you need? So in oncology, for example, with panel sequencing, it's like ask for only what you can deal with now, and if you want more later, then go get it later. I mean, it's an interesting cross-section here because we have interesting different sizes of genomes. Well, they're all basically mammalian except for the trees, but you know what I mean.
Trees are people too, come on.
I'll go first and just say that this is a problem for us. We're actually going for a limited panel, but when we say limited, you start digging and suddenly it becomes unlimited. And Corey and I were just chatting before coming into the meeting, and I said, actually, this is this week's problem, is where we stop on the SNP selection process. There's a cost restriction for us at the moment, so we're not going to go and look at a lot of SNPs that are related to other things. We also don't want to get into the prognosis and prediction game. So I don't want to be answering patients who are saying, and could you tell me whether I'm going to get breast cancer? Now you can tell me whether I can react badly to this drug. So I'm cautious about getting into that. That's 2 years, I think, away in primary care. So I might expand it for the drugs and pharmacogenomics, but I really don't want to get into prediction and risk of disease at this stage.
To address your question, I don't know I don't know the answer. I know that we'll very quickly get into a situation where we have too much data to even physically wrangle, let alone analyze. But because of the nature of the two-stage process of our project, we're actually collecting whole exome data and benchmarking it against a 18 SNP test. So we'll know the quality of the exome data, and we'll then be able to requery that data for additional drugs. We're starting with a single drug, but we'll be able to requery with multiple drugs. Whether that's a sustainable, scalable solution, we really don't know, but we're going to take the empirical approach. And whole exomes will we selected based on cost. By the end of the project, whole genomes will be the same cost. So it's a very fluid thing.
Well, not being a scientist, I probably can't answer your question fully, but I'll refer you to Pia Smets in the audience for details. But I do know that from the work that has been done, that the genomes for trees are very, very large, referred to and bloated. So there are only a few genes, of course, of the thousands of the millions of SNPs potentially that are responsible for specific traits. So trying to narrow it the analysis there. But what I understand is the computing power of the technology has advanced quite rapidly over the last several years, and so the ability to store large amounts of data it's not as much of an issue today as perhaps it's in the past, and nor is it as expensive to test. So at this point, I'm not aware of any capacity issues. But again, refer you to my colleague here for details.
Yeah, I just want to re-emphasize the idea that 3 years from now, we may go to a store-nothing approach because it may be cheaper to resequence as you need it. So it'll be very interesting to see how things develop. We don't want to use up all of the electrons in the universe.
So Brian and Marty, you presented really compelling economic arguments why you would use the technologies that you both discussed. So Martin and Corey, I'd like you to comment though. So if your technology, if you validate your approach in primary care or at the community pharmacy, that's not going to be enough. I mean, there's still going to be reimbursement. There's still going to be regulatory hurdles. Can you comment on that? I mean, how, once you go through your first gig, even your second gig, phase 2 of the project, I mean, you still have that final hurdle of somebody has to pay for that technology. And how you anticipate that happening, and how you anticipate having the approval for that being, I'll call that the regulatory hurdle, and I'm using that term broadly.
Okay. Just leave the hard one for me. That is ultimately, if no one pays, it's not going to happen. And so right now, if we look at the U.S., there are there's a handful of drugs for which pharmacogenomics is strongly recommended in the label, on the labeling information. But there's almost 200 drug-gene interactions for which there's good evidence in literature, but no recommendation. And so what we have to do is move the needle closer to 200 from 10 right now and demonstrate using all of the power of pharmacoeconomics that we have at our disposal to show an economic benefit. That's again easy to say. The second part of that equation is finding out who the stakeholders are and how interested they are in being early adopters. And so our partner, I know, is engaged in a very extensive effort to interview stakeholders. And when you talk about complementarity, Martin just mentioned that he finished his interview just the other day with the firm that's doing the stakeholder engagement. So that's the large third phase.
We talked to a group in Indiana, Dave Flockhart's group, who are doing a multiple SNP, multiple disease approach, but more hospital-based for internal medicine. And they were demonstrating the economic benefits, and this was about 18 months to 2 years ago. And that was one of the triggers that actually said, well, if we do it in primary care, we should have similar sorts of economic benefits, if not more. The advantage of doing a multiple disease approach is that you can demonstrate the economic benefit far more. We've got good, reasonable, it's high-level evidence, but it's a reasonable benefit for just you looking at warfarin alone. But if you take what we're doing is multiple diseases, 26 diseases, it's not because there's a SNP for every drug used for those diseases. There's probably a SNP for one drug that's used in those 26 diseases. And so we are working with health economists to actually demonstrate that. We produced figures, and you can go from saving Canada 10 billion to 1 billion a year, because the scope at the moment of uncertainty is quite large. What we hope to do with the evidence we'll have in a year or so is reduce that uncertainty and be able to say, okay, if you introduce routine SNP testing, it will be 1.6 billion for Canada. So absolutely right, we've got to have that evidence, and the payers have to have that before they'll start paying for the tests.
Please.
Yes,
we'll get you a microphone here.
So, question for Marty. So you're talking about the milk production. So I'm thinking that probably are you also following like many other genes, like a panel of genes, for example, genes that may affect like a disease resistance, the cow may produce more milk, but is more susceptible to diseases?
Well, there are other people who are doing the research. I'm using a commercially available test that tests about 12,000 SNPs on a low-density panel, a high-density panel, so 68,000 SNPs. There's a group out of Ontario who've been doing what they call the genomics of immunity, and they've identified some bulls that have a better immunity or a better primary and secondary response to vaccinations, for instance. And they're promoting that as an immune plus right now in the genetic line being sold to the farmers. So yeah, as we get more information, and with UBC was one of my testers, by the way, in the UBC dairy, which is out of Anglesey, they're being involved in actually bringing that testing into BC, and they're being one of the herds to be doing that. Wouldn't it be nice to be able to do it all in one hair test? And that's the gold standard, but there's also intellectual properties that has to be dealt with.
Any other questions? Okay, if not, I'd ask you to help me thank all our speakers. Greatly appreciate it.